Think I figured out my iPad today, folks. So are you ready? I can actually give you some notes. Okay, let me get the chat visible over here so I can see what you're all saying. Let me mute myself so you don't get a crazy feedback loop. I've got, for some reason, my lighting in the corner has just gone out over there. It's a little bit dark. Hey, Megan. Okay, so let me know, are you in the exambulance? Have you registered inside the exambulance is something I want to check in with you first about, because we're going to be making some changes to the things we're doing. We've just had crazy meetings this week. Well, I mean, crazy meeting is just madness right now. If you're on Gibber and you're not in the exambulance, then tailoredtutors.co.uk, we're actually going to create a page for it itself, but right now there are buttons on the home page to sign into the biology, the chemistry, maths, study at home exambulance. So it's going to be there to give you structure and to support you while studying from home, whether you're a year 12 student or you're a year 13 student. So it's basically going to give you, it's a way to structure. We're going to be doing lots of live classes to support it. One of the main announcements is that we're going to be moving these lives from YouTube, and we're going to be doing them inside. You'll need to be inside the exambulance to do it. It's just a bit glitchy using YouTube, and we want to support the students inside the academy, because then we can link you to things much more easily and support you. So we're still going to be available for lives, but it won't be on YouTube Live. It'll be inside of the exambulance, totally free. You can register from the Tailored Tutors website. There should be a link in the description. So that's a link to our homepage right now. The buttons to register are on the homepage. As I said, I think we're probably just going to create a page to describe what the study at home exambulance is a little bit better. Yep, there's one for biology, there's one for chemistry, one for maths, and that's going to be the same. We're going to complete this week's worth of lives on YouTube, and then we're going to move them inside of the course. What else was I going to say? Any other sort of general bits and bobs from me? Today we're going to do I'm sort of a day behind this week if you're following the schedule that's in the exambulance, because my internet crapped out on me on Monday. So I will look to make that session up, whether I do that this week or next week. If you are going to be joining us in those live classes next week, then you'll need Zoom. It's a very, I'm sure everyone's pretty familiar with Zoom right now. Lots of people catching up with each other on Zoom. So it's replaced the pub to some extent. It's a super lightweight plugin that you can download. Any questions about the exambulance? House party is not going to be great for teaching, to be honest with you. Any questions about how to use the exambulance, where to find it, how are you using it very much right now? Obviously, it's got all the MID sheets in there, the Markside drop sheets to help you support you with your past papers, and we're sort of moving into doing some past paper practice in a couple of weeks. And obviously completing the content for year 12s is going to be the major piece. If you're really like, the stuff that we're doing with you in these live sessions is going to be useful, but if you really want to finish the content, then the content guide is going to be the only real source of like everything information for you. Do I think the year 12 exams are going to take place this year? No, but next year, the A levels, as we're assuming, everyone's assuming, nobody really knows. Obviously, it depends on what happens, but almost certainly the exams will happen next year. I mean, I think 12 months down the line, things should be back under control a little bit. So yeah, for sure. But in terms of the AS exams, there'll be no examinations in May or June this year. There will be, whether they'll do the AS exams in the autumn, September time. So again, just to update you, we're going to make an announcement on Friday about how the A-level grades will be determined, if we're allowed to. We are waiting for the government to officially announce it so that we can tell you. We have a pretty good idea, we've been told, but we're not allowed to say anything until then. So I'm not going to tell you before then, but come back to the YouTube channel. We're going to set up a live for Friday, and assuming that we're allowed to tell you, we'll tell you as soon as we possibly can. And that will give you a bit more information, you year 13s, about how to use the, how you can prepare. But basically, you're going to get given a calculated grade. And if you don't like your calculated grade, then you can do the exam in the autumn, which we're pretty sure is going to be in September. So if you don't like what they give you, then you've got a chance of coming back to it. But what I don't think is a good idea is for you to just do nothing until they give you a calculated grade, because then you won't have enough time to actually get anything to improve your grade. You'll be like, I'm better than a C. But then if you do no work between now and the end of July, and then you sit an exam, you're probably going to get a C or a D. So don't shoot yourself in the foot. Exam guide and content guide for year 12. If you want either or on their own, I think it's £39 a month, including all the live classes and asking us questions and all the content, obviously. If you want them together, you want both of them, and that we call that the ultimate guide, then for AS students, I think it's $59 a month. Actually, I don't know, which is pretty shocking, but it's all on there's a pricing page on the website. It's all pretty transparent. Okay. Let's jump in. Let's do some protein synthesis. So we yesterday looked at sort of DNA, genes, chromosomes, how they're organized and folded, differences between DNA and prokaryotes and eukaryotes. And now we're going to be looking at how how proteins are made, basically, how the DNA actually codes for the protein. So I'm going to be doing every day until Friday, and then we're going to be doing every day next week, but they'll be inside the Exambulance course. You'll need to be inside the course to get it into the live. It's just going to be way easier for us to teach you in there. Okay, let's get back to where I want to be. I'll just share with you. I think I figured out screen sharing now. I'm going to share my laptop screen with you first of all. So this is the AS study schedule inside the Exambulance. You can see we're doing study with me's live every day as well right now. So we're just experimenting, doing a few different things. We are currently here. I'm going to be doing this session today. I'll try and get this document updated. It's not very high on my list of priorities though, to be honest, because I've got lots of different things. If you want to check out the AQA, the OCR, and the Edexcel content guides, there's buttons here to go and just have a look at them and see what they look like if you're not one of our subscribers already. If you are a subscriber, then it will obviously just take you into the content guide. Okay, now let's change my screen sharing. This is, I'm doing it in a new way. Okay, so I'm going to do this and then this and then this. And we're good. Oh, look how quick that is. Oh, do I have to click start? Yeah, I love it. No cables as well. I'm very happy. And on my new computer, I've got a much bigger screen. Okay, let's to the front, preferably where zoom. Okay. I am super confused by what you're seeing right now. Doing new things all the time. Okay, so we're going to start off with a bit of transcription. What is transcription? What do we do transcription for? What's the purpose of it? Covered some of this already, but I want to see what you're seeing a little bit. Okay, so I'm going to start with the diagram actually. So DNA, I'm going to draw as, we've got, how much of the page do I want to do? This is all taking place inside the nuclear membrane. So this, I suppose, would be our, we've got our pores in our nuclear membrane, like this. So this is our nuclear envelope. It would, I guess, carry on like this. We've got our DNA. It's forming our double helix. I'm going to draw this hand branch, sort of, I'm just going to draw that one. I'm not going to draw it. I'm going to draw it as a dotted line. It's there, but I'm just not going to draw it because it's going to get in the way. And we've got our DNA template strand here. So we've got our enzyme. What enzyme is going to open up the DNA? It's to break the hydrogen bonds. I'm going to draw it as a triangle, like it's like probably best to call it DNA helicase, but helicase would probably get away with it. So this is going to be DNA helicase. We've got our DNA template strand because it's the template to make the messenger RNA. And we've got some bases on here. So I'm going to say, I'm going to do T, let's do T, A, C, G. Okay, and these are, now that we've broken the hydrogen bonds, these are unpaired bases. Yeah, normally their anti-parallel complementary base would be on the other strand, which I've just sort of drawn as this dotted line as like it's tailing away. So that's unpaired. what's going to come in? Well, we're going to have free-floating RNA nucleotides. So obviously a nucleotide is going to be our ribose, because this is RNA. We're going to have our nitrogenous base here, which is going to be either our A, U, C, or G, one of either or. And we've got our phosphate group. So that's our nucleotide. I'm going to draw this as like just a T shape. And let's say this is an adenine base. I was going to just represent this nucleotide as like a shape like this. So we're going to form our A is complementary with our T. And this is this base. And so we're going to basically form our bases. A is going to pair with U. Remember, there's no thymine. C with G. If you can, maybe if I zoom in a little bit here. And let's say this base here, G is going to partner with C. But this one I've not drawn connected. I've not drawn this connected because what's the hydrogen bonds will be forming here. So we've got actually 3 hydrogen bonds forming between C and G, and there'd be 2 between A and U and T and A. But it's not connected to the polymer yet. So what enzyme is going to catalyze this reaction to join on each RNA nucleotide as we go? So this is going to be another enzyme here. This is RNA polymerase. And this is catalyzing the reaction to form what bond? What bond are we forming here? And then we'd have maybe this A base would just be sort of arriving on its way to get paired up. It's forming a phosphodiester bond. So here would be a phosphodiester bond, here would be a phosphodiester bond, just where the nucleotides are connected together. And the RNA polymerase catalyzes the reaction that makes that phosphodiester bond. These here are hydrogen bonds. Okay, so that's basically what's going on if we were to zoom right in. We're then going to basically make an RNA copy. Yeah, H bonds are the bonds between nucleotides on the opposite strands. The bonds between nucleotides in the sugar phosphate backbone are phosphodiester bonds. So we're going to make a complete copy of the template strand. And this is going to include all of the bases. So there's basically, I'm going to do the red bits here. How much space have I got? Something like that. I can use green, why not? And so we've got some red, some green. And some of these code for the proteins, some of these don't code for the amino acids that make up the protein. The free nucleotides are just floating around inside the nucleus. There is an abundance of them there. They're small, they can fit through the nuclear pore. Yeah, in the nucleoplasm. Can I circle a phosphodiester bond? Well, I put them as dots on here. It's the bond between, if we had our A here and our T here, oh, well, it's another A, then the phosphodiester bond forms here. I'm gonna delete that, but I suppose if I were to draw it, let me draw it in red, it would be connecting to this bit here, and this would be our here. That makes sense? I think that's pretty much, I just don't want to pollute my drawing too much. So here we have exons, and we also have introns. So this is a complete copy of everything that's on the template strand. And it contains both introns and exons. What process is going to take place? This is all happening still inside the nucleus. Remember, we've got all our nuclear pores down here. I've drawn this quite nice and small. I thought I was going to be really tight for space. We call this molecule that includes the introns. If you're AQA or EdXL, you're going to call this pre-mRNA. And if you're OCR, you're going to call it primary mRNA. And it's going to undergo the process of splicing, which again is also a key term, which is the removal of introns. Well, maybe I can put that. The process of removal of introns from the pre or primary. I'm going to write pre because it's what most of you have to call it. Or N, if I could write letters, RNA. And this forms basically, we're left with only the exons are exported. So it's going to be shorter because it's less, that's probably a bit too short. It's going to be shorter than our original piece because it's lost some bases. The introns have been removed. And this is converted. Actually, I kind of want to remove I wonder if I can move things. I don't think I can. I'm going to just delete it because I want to draw an arrow. Pre or primary messenger RNA is converted into just messenger RNA. And this is splicing. Maybe I can fit splicing in here. Okay, it's probably not needed. Any questions there? It seems like everyone's kind of on top of things. So some of you, it seems like you've done this topic. I mean, so transcription, translation is, I'm picking it because I'm assuming some of you might not have done it. Pre-messenger RNA is the messenger RNA that's the direct product after the DNA has been transcribed. So this is the transcription process. And if you think about what does a scribe do? What is a scribe? Old school word for to write, yeah, to copy. Yes, but it's to make, like a scribe would maybe copy, they'd literally handwrite new versions of the Bible because before the printing press, somebody had to write it. So a scribe would just kind of copy things out, but it's just copying. So we're going from DNA based to RNA based, but you're still saying nucleotide to nucleotide. This is still made of a polynucleotide strand. It's just RNA rather than DNA. And there's a few differences, like the sugar is not deoxyribose, it's ribose. So there's a few things that are a little bit different, but not a huge amount. Whereas when we're going to change it in translation, the next stage, we're going to go from a polynucleotide into an amino acid sequence. And so that's a different language. We're moving from polynucleotide to polypeptide, and therefore it's a translation. You're converting the language of nucleotides into the language of amino acids. And obviously this has got all our bases on it. I mean, if I were to copy these across, if I were to take the ones we've got over here, I'm just going to, oh, that's not what I wanted to do. I wanted my pen back. So let's do them more spaced out than that. So this, if I'm just copying over, I've got to have A, U, G, C. And obviously I've only just put a few in there, but I'm just copying these guys over there. And that's where those letters come from. What acts as the scribe? Well, the RNA is a copy of the DNA template strand. I suppose RNA polymerase itself would be the thing that is making the new strand. So I suppose that would be why is it important? Because you own the protein needs to be functional. It needs to have the correct tertiary structure. Therefore, it must have the correct primary structure, which means it must have the correct messenger RNA codon sequence, which means, but the DNA contains introns. Why does it contain introns is a question I'm not sorry I can answer, but it's important that the mRNA, the finished, mature messenger RNA, if you're an OCR student or just the mRNA student, post-splicing, is the correct sequence of codons to code for the primary structure of the protein. Why do they exist in the first place? I don't exactly know. Okay, so what can we say here? Let's use some of the key terms we've got on the page. We can say DNA helicase breaks the hydrogen bonds or separates the strand. I'm going to say separates the strands or unzips the DNA. You can actually say unzips the DNA double strand by breaking the hydrogen bonds. Wish it was easier to change colors on these things. H bonds. You should write hydrogen bonds in your exam. I'm just using shorthand because I'm short on time. The free RNA nucleotides attached to the template strand. RNA nucleotides. It's supposed to say template strand by complementary base pairing. There's nearly always a mark for that. Whenever you're talking about bases pairing up with each other, you want to include that phrase, complementary base pairing. We can say RNA polymerase whilst I'm in red. Joins the nucleotides together or forms the phosphodiester bond is probably best. Let's to form pre-mRNA. Why can't I write an N today? Or if you're OCR, it's primary. Okay, what did we learn yesterday about the differences between prokaryotic DNA and eukaryotic DNA? No, only one strand acts as the template. That's one of the main differences between DNA replication and transcription. They're very similar processes. Only one strand acts as the template strand. Maybe we could, I wonder if I can squeeze that in. Only one strand acts. So if you've got compare and contrast the processes of transcription with DNA synthesis, then you could say that as the template. You could say only one strand of messenger RNA is formed, whereas obviously you're going to get two strands of DNA in semi-conservative. Okay, so the other DNA strand that's actually the sense strand, it's called, is left just unpaired. You actually get a bubble, is what you get, like a little replication bubble, but we don't need to go into those details. The template strand is the antisense strand. Okay. Okay, so we've got this process here of splicing, but in prokaryotes, we basically go from straight into DNA. Transcription produces the messenger RNA directly. What am I doing? I can't write N's today because there's no introns. So therefore, there's no splicing. Whereas in eukaryotes, we go DNA to pre-mRNA. Obviously, if you're OCR, it's primary mRNA, and then we produce mRNA, or if you're OCR, mature mRNA. So the introns are removed. And then I suppose one thing I haven't put on here is that our messenger RNA, once it's formed, comes into the cytoplasm for the next stage, which is translation. And this is our nuclear membrane. I actually fit that in way smaller than I was expecting. Nuclear envelope. And this is our nuclear pore. Questions like, why do prokaryotes not have introns? I don't actually know, to be honest with you. If you want to go research it and let me know in the comments of the video, then I'd love to know. But that tells you that it's not something you are going to be asked, not even as a suggest question. Okay, let's jump across now to a bit of translation. So the next stage here, just checking that you are, yeah, everything still looks like it's working. So what is I'm going to start with a couple of key terms. How long we got for time? Oh, we're not doing too badly. What's a codon? And what's an anticodon? What's the difference here? Okay, so it's a sequence of 3 bases that codes for a specific amino acid, and it's on messenger RNA. You could say 3 RNAs, lots of different ways you can phrase that. And an anticodon is a triplet of 3 bases. That is complementary to a codon. I'm going to say 3 bases on tRNA, transfer RNA, that is complementary to a codon. To an mRNA codon. Okay, let's just take a little look at tRNA here for a second. Single-stranded or double-stranded transfer RNA? After specific amino acid on messenger RNA is what that says right there. I could probably tidy that up a little bit for you. Single-stranded or double-stranded for transfer RNA? Single-stranded, containing hydrogen bonds or not containing hydrogen bonds? Compare and contrast the structure of messenger RNA and transfer RNA is a question that does pop up from time to time. So it does contain hydrogen bonds. So we'd get maybe some bonding in here. So this is what causes it to be this folded weird shape. It's only one line, it's single-stranded, but it's folded into this shape. And it has a specific anticodon and it carries a specific amino acid. So this would be our amino acid. And its anticodon, which I have failed to draw, would be its 3 bases on the end down here. And this would be our, let's do it and label it in black, our anticodon. And whatever we have here, the base, the anticodon, is specific to the amino acid. So it's I was going to say it's a polynucleotide. Yeah, it's a polymer of RNA bases. It's folded by hydrogen bonds. The anticodon is specific to the amino acid that it carries. We're not talking about a protein here, so primary, secondary, tertiary, quaternary doesn't apply. This is a polynucleotide, not a polypeptide. These are our hydrogen bonds here, which is why it's not just a flat line. So messenger RNA, single-stranded polynucleotide strand, no hydrogen bonding, therefore it's a straight line. So let's draw a strand. How am I? Okay, so again, we've got this is our mature messenger RNA. I've copied the red over from the previous ones. We're going to start with a start codon. And then I'm going to draw them spaced out. Obviously, they are continuous. We're going to have our codons in sets of 3. Where am I going to draw? I'm going to draw I might have to sketch this a few times. So I'm going to draw my ribosome fairly fat. I think I'm going to start all of that again. I think I need a little bit more space below it. In fact, let me draw my ribosome first. Okay. In fact, we can do our start codon. And then I'm going to do 2 codons. In each ribosome can hold 2 transfer RNAs in position at once. So CGU and AUC on our messenger RNA, which is going to be complementary to C. And let's actually get in a bit closer. GC AUAG. Oh, now I've gone and drawn it. Wish I could move things more easily. Now I don't have enough space above my drawing, but I reckon I can squeeze it in. I know what I can do. What happens if I just do that? I'm just going to have another crack at my ribosome. Just a slightly weirdly shaped ribosome now. I reckon I can do that probably slightly better. Something a bit like that. Okay, that will do. How many can I draw in here? So I've got okay, so if I draw this as codon 1 or 2, 3, then this will be amino acid 1, 2, 3. Because the start codon doesn't count. That's maybe. Why does the messenger RNA become single-stranded? Well, okay, the DNA stays in the DNA. The DNA polynucleotide doesn't ever leave the nucleus. You're making an mRNA copy of the template strand, and then that single-stranded copy then leaves into, it's the messenger RNA. It takes the message of the DNA from the nucleus into the cytoplasm to be translated. And so these are our amino acids. How are the amino acids joined together? I'm going to draw them with a red. And then this is catalyzing the reaction right now. This is why you hold two in place at the same time, because we're forming this bond right here. And it's important that there's two transfer RNAs next to each other, so the two amino acids are next to each other, so you can bond them together. And we're forming peptide bonds in a condensation reaction. Let's label it in black. What am I missing? Well, I suppose my anticodons are not attached to my transfer RNAs. This is forming a polypeptide chain. And this is our ribosome, which can hold two transfer RNAs in place at the same time. And then we'd obviously have another, the next codon, I'm just going to, is here. Let's say this is GUCUG. And the next transfer RNA is not inside the ribosome yet, but it's going to start sort of, it's going to be on its way over. Sort of swooping its way in, which is going to be GAC. And it's going to have its messenger RNA. It's going to have its amino acid attached to it. However, the transfer RNA that's just done, I'm going to draw one over here. We're going to have a transfer RNA. I have to draw it a little bit smaller because I'm running out of space. But this transfer RNA that's just contributed, let's say that this one, we've got amino acid 3. I'm just going to call this transfer RNA 3. This one doesn't have an amino acid attached anymore. This one is going to go back into the cytoplasm and go pick up another loose amino acid from the cytoplasm, and then it will come back around and be recycled. But this one is given its, in fact, it's probably better to put that arrow underneath than on top. It's given away its amino acid. Its amino acid is, it's this one here. It's already, this was the previous one to attach, and therefore it's already given it. These are codons. I think that's pretty self. Any more questions about that? Let me take a look at the chat. I'll put some notes on the page. This is taking place in the cytoplasm. Yes, correct. Oh, what direction do the ribosomes move in? I don't know, which tells you that they don't ask. I have never in the history of A-level biology ever seen a question about that. I would assume the unpaired bases, I could work it out, but it would take me a little bit of time to figure out which bases, because you've only got unpaired bases on one side, right? You've got the sugar phosphate backbone with the nucleotides unpaired on one side. I think it is the 5' to 3'. Does someone want to Google it? Someone can Google it, but they won't ask you, so don't stress. Good question, Calvin. Kelvin. Why are there different codons to those of messenger RNA strand made in the ribosome? Megan, can you rephrase? Why is there different codons to those of the messenger RNA strand made in the ribosome? So in this diagram, the ribosome is moving in this direction. You'd have a stop codon on the end as well. It is 5' to 3'. Oh, and Helicase do it. Interesting. If in doubt, 5' to 3'. Big to small. Does the mRNA translate into tRNA? No, the messenger RNA translates into it's a translation. You're translating the language, the language of nucleotides into the language of amino acids. So the messenger RNA is translated to become the polypeptide chain. The sequence of codons determines the sequence of amino acids in the polypeptide chain. The transfer RNA is just a bit of machinery that is used. Do you need to know how to draw an amino acid structure? Yes, you do. And know how to, and obviously I've drawn them as blobs here, but amino acid structure, you've got a central carbon, and then you've got, well, in fact, let's do with a carbon makes 4 bonds. So the central carbon is surrounded by 4 bonds. You've got an R group on the top, which is different between all the different ones. You've got a hydrogen group opposite that, and then you've got 2 functional groups. You've got the carboxylic acid group or the carboxyl group, and you've got the amine group, which is NH2. So that's how you draw an amino acid. And obviously this bit is the bit that's different. All other amino acids have this in common, and the R group is the bit that's different. The simplest possible one is glycine, which has just got a hydrogen here. So that's glycine. It's the only one that I can actually draw because I don't know any of the other side groups. But you would be expected to draw that, yes, or to be able to draw that, and call it the carboxyl group and the amine group, not the amino group. You can call it the carboxylic acid group if you so choose as well. I'll just put some notes down and then I'll come back to questions, I think, at the end. So ribosome, I'm going to say the ribosome binds to the mRNA, maybe at the start codon. The codon is matched or paired to an anticodon by complementary base pairing. The ribosome holds the 2 transfer RNAs in place. So the amino acids, so the 2 amino acids can be joined by a peptide bond. Does actually require ATP to make that happen. Never seen a question on it. Amino acids are joined by condensation to form peptide bond. And this forms a polynucleotide chain, or our primary structure. To form the peptide bond is what's going to require ATP. The ribosome is usually attached to the rough endoplasmic reticulum, which is attached to the outside of the nucleus in the cytoplasm. So it's taking the general location is the cytoplasm, or basically not inside the nucleus, because transcription happens in the nucleus, translation in the cytoplasm. The site of protein synthesis would be a ribosome, and often the mark schemes will accept the rough endoplasmic reticulum, because usually those ribosomes are attached to the membrane of the rough endoplasmic reticulum. The codon is the sequence of 3 bases on the messenger RNA that code for, how can I do 3? That are complementary to the 3 anticodons on the transfer RNA, and the transfer RNA attaches to the amino acid. So the codon determines which amino acid gets added. But it's not, the codon is not the same as an amino acid. The codon codes, it gives the instruction for which amino acid should be added in which order. So it will go, amino acid number 1 needs to be glycine. Amino acid number 2 needs to be valine. Amino acid number 3 needs to be lysine. And so the sequence of codons on the messenger RNA determines the sequence of anticodons, and the anticodons are specific to the amino acid. And so the codon codes for which amino acid gets added in which order. And that determines our primary sequence, or primary structure of our protein. And obviously the primary structure is going to organize the sequence of the R groups, and those R groups are going to interact in different ways to fold the protein into a different 3-dimensional structure. Do you need to know the structure? Only, Jacqueline, the structure that I put on the page. It's a polynucleotide, it's folded, it's single-stranded, it contains hydrogen bonds, it has an anticodon that's complementary to the codon, and its anticodon determines which specific amino acid that it can carry. Each tRNA can only carry one type of amino acid and has one anticodon. That doesn't change. So that's the, again, always in my notes in the course or the things I'm teaching you, the detail that I'm teaching you is the detail you need to know. So do you need to know the structure of tRNA? Yes, but only the stuff that I put on the page. Yes, so always, if you've got nucleotides forming complementary base pairing, always by hydrogen bonding. A knows to pair with U or T because it forms 2 amino acids together. C knows to pair with G because it's going to form 3 hydrogen bonds together. Did I say amino acids? So that complementary basing is controlled. Complementary base pairing is controlled by hydrogen bonding. tRNAs are in the cytoplasm. Do you need to know which enzyme forms the peptide bond? No. Well, basically, it's the ribosome is going to help catalyze that reaction. There's no specific enzyme that you need to recall for that. The ribosome is involved in forming the peptide bond. Again, it's going to hold the amino acids. It's going to hold the carboxylic acid group of one amino acid next to the amine group of the next amino acid so that they're in the right position. It's going to help basically form the bond. If in doubt, say that the ribosome, I think you probably could get away with saying it catalyzes that reaction. Whether that's technically correct or not, I don't know. It's not in the specs. So you're not required to know. It would be maybe a suggest question. Okay. Does the hydrogen bond reform when the RNA nucleotide pairs with the DNA nucleotides? So you're talking about transcription there, Anna? Yes. The hydrogen bonds form between the template strand and the RNA polymerase, messenger RNA or pre-messenger RNA strand that's forming inside the nucleus. But then as soon as the messenger RNA strand is complete, it breaks those hydrogen bonds and it actually goes and gets spliced, and then it leaves the nucleus. And then the other strand of DNA will come back in, and DNA will just reform its regular anti-parallel structure. Peptide bonds are the bonds formed between the amino acids to form the primary structure of the peptide, the polypeptide chain or the protein. You don't need to know how transfer RNAs are made. Note what function does the polypeptide chain have? It's a protein. That's it. So the polypeptide chains are proteins, depending on whether it's a quaternary structure or not. If it's not a quaternary structure, the polypeptide chain is the protein. If it's a quaternary structure protein, it's got more than one. So you'll then assemble multiple different polypeptide chains together to form a larger protein, globular protein. In fact, does it have to be globular? No, it doesn't, because collagen's a quaternary protein with a quaternary structure that's got it's fibrous. There is a lot of AO1. The trouble is, this is so fundamental. It is a common AO1 question, but you have to understand how DNA ends up, the sequence of DNA makes the structure of a protein. Like that knowledge is examined left, right, center, up, down, AO1, AO2, not so much AO3, but like the principle of understanding how you go from the DNA sequence into the primary and therefore the tertiary structure of a protein is absolutely key and fundamental. In terms of marks on the exam, in terms of examining this, I'd say 50% AO1, 50% application, but in terms of your understanding its core to how it all works. Grace, I'd say there's a good chance you won't be going back to classes until September, but we don't know. Nobody knows, but there'll be links to the Zooms for next week inside the Exambulance. So just again, we're going to be not doing our broadcast onto YouTube, or maybe we could broadcast them to YouTube, but there'll be no one looking at the chat in terms of joining into the joining in and asking me questions like you're doing now. Then we'll be available in the Zoom room. It'll be way more responsive. I have to wait like 5 or 6 seconds for you guys to give me an answer when I ask you a question. Yeah, you could say I'd say the codon and the anticodon, they're both nucleotides. They join by complementary base pairing, hydrogen bonds. Okay, we're going to be making an announcement about how your grades, or in fact, year 12 students, you're probably less interested in this, but for year 13s, about how the grades will actually be calculated on Friday, if we can. Okay, I'm basically done here. I think I've answered. Yeah, okay. In fact, the only live class I didn't finish from my regular series because of all of this happened was my class on the practical skills. So I'll be doing that inside the course for my regular subscribers pretty soon, Elizabeth. So do you have a subscription? If you've got a subscription, I'll be doing that. I might not do it this week because I'm still basically catching up on coming back to the UK. But yeah, I will be doing a live class on that and catching up with my students in there. We don't know. We'll announce it as soon as we can in the afternoon, basically. But check out the YouTube channel. We'll create a live event and schedule it for Friday. Probably, I'm not committed to anything here, but ish 4 o'clock, I would think, something like that. We should know by then, or we know already, but we should be able to tell you by then. tailoredtutors.co.uk. You can, or if are you in the Exambulance? In the Exambulance, you can go sort of explore. But if you want to start a subscription, then tailoredtutors.co.uk. All the pricing is there. If you're a year 12 student, all the AS content is there for you to study everything from home in all those videos. We're going to be doing not Saturdays and Sundays. We're just experimenting right now, to be honest with you. Like, we will be doing next week. We're going to do lives every day. Biology times will be in the afternoons next week. So just keep your eyes on the Exambulance. We'll be updating it with new video and new content, and adding a live class registration page. In fact, you won't really have to register. You just like go there to join the Zoom, join the live class at the time. So you won't be able to access it from YouTube. You'll have to access it from inside the Exambulance. I don't think it'll be a registration, Jacqueline. You'll just go in and then like join the live at the time. Just go in. You won't have to register in advance. I think we'll just open it up to anyone inside the Exambulance. You will need the exam. If you want to join the live classes, then you'll be needing Zoom, because it will be all inside of a Zoom room together rather than being on YouTube. It's just going to be way easier for me to teach, because I can ask a question, get an answer, ask a question, get an answer, as opposed to ask a question, fill some time, wait 5 or 6 seconds, and then pick an answer. I don't know what I'll be doing in terms of topics next week. I will be doing lots of stuff to support you around studying from home and scheduling your time and doing drop-in questions and stuff like that. So in fact, I've got 5 minutes before I go start my next class. I need to close this Zoom room down. I need to go set myself up and get my notes ready. So I will see you tomorrow. Same time, same place. Let me go find the Zoom control, the YouTube live control room, because otherwise I can't end the stream. Okay, this is it. See you later.